Great. Thanks so much, Carlos. And I'd just echo Yelta's comments on how fruitful the exchange back and forth between the European AFM group and our group has been. It was just a total honor and pleasure to host Yelta and have him over to see what we're doing. As most of the folks on the call know, the history of this was that Europe really focused on EV68-based surveillance, whereas the US focused on AFM-based surveillance. And now we're kind of learning from each other. But I think back to Matt Elrich's question, it's very hard to get at the total burden of EVD68 respiratory disease here in the US. We have this CDC sites, which are doing great work, but it's kind of sentinel site surveillance. But it's very hard to grasp the overall burden, across the country. Whereas in the countries in Europe that are doing EVD68 surveillance, they are doing very widespread surveillance at concentrated clinical virology labs. So they have a better sense of actual burden of disease there. And they're working on building up their AFM surveillance, which was part of the reason for Yelta coming over to visit. So it'll be interesting to see as both places kind of beef up the other side of their surveillance, if we could better answer those questions. So this is a bit of a side note to end our conference today. Carlos asked me to just give an update from the kind of PEDS ID perspective of what's going on with other respiratory viruses in this very unusual year, with this extended surge, which started with EVD68, as Sarah noted, and now has moved to other respiratory viruses. So I just wanted to start out, this was one of the first captures that I had of that BioFire trend data back during the lockdown period of the pandemic in March of 2020. And this is just percent positivity off of one of the most common commercial multiplex respiratory platforms. And what you see is just a drastic dive in the percent positivity, and just shows how effective the non-pharmaceutical interventions that were put into play at that time were, not only for SARS-CoV-2, but also for our typical childhood endemic respiratory viruses like RSV, influenza, HMPV, adenovirus. And you can see all those dive, and that led a lot of us wondering what was going to happen when they came back. And as the NPIs started to lift, you saw some unusual patterns starting to emerge. The first virus that came back were the rhinoenteroviruses, mostly rhinoviruses here with the red line. And then you started to see, as Yelta mentioned, non-seasonal emergence of viruses that typically came during very well-defined seasons. So RSV and influenza are wintertime viruses. We saw those RSV emerge in the spring through the summer and fall of 2021, and then a small blip of influenza starting out of season as well that year. So we did some modeling, mostly for the EV68 question. This was one of our fellows, Hai Win Tran, who worked with Brian Grenfeld's group, and Daniel Park, who've been on this call and are part of the AFM working group. But they had done the previous modeling showing that the outbreaks in 2014, 16, and 18 were likely driven by the growth of a susceptible birth cohort, meaning enough children had to be born that hadn't seen the virus, that wouldn't have had immunity for the virus to circulate again. And that periodicity took about 2 years to build up again before it could circulate again, and had predicted that obviously we should have seen a large outbreak in 2020, but lo and behold, we did not, likely due to those MPI measures that we had discussed earlier. So then the next question was, what was going to happen after this kind of honeymoon period during the pandemic? And what they modeled was that we would expect to have a larger susceptible population that did not have recent exposure to EV68, and that when it recirculated again, there was the potential for a larger outbreak, as you can see in that figure on the right. And I think that really is what we indeed saw this year with the large, widespread EV68 respiratory disease outbreak. So then we took that a step further and said, what about the other respiratory viruses, influenza, RSV, that come every year during that season? And we did some modeling in this Lancet paper with that same group. On the superior part of the y-axis is the previous kind of seasonal circulation of endemic respiratory viruses. You could say this is RSV, influenza, whatever. And the inferior y-axis is trying to model that susceptible population. So new births of children who wouldn't be expected to have immunity. Blue is younger children, red is getting into older children. And really just trying to show that if you have a period where the virus doesn't circulate for a particular period of time, that susceptible population is going to grow even bigger and is also going to be older. So if it was RSV and you used to see disease in kids under 2 years of age, and you don't see circulation significantly for 2 years. That may be kids under 4 years of age. And then when you see the outbreak, it could be a larger spike in a shorter period of time, including older kids. And I think that's part of what we're seeing right now with the significant resurgence of RSV that all of us are experiencing at our children's hospitals throughout the country. So back to that BioFire syndromic trends. Again, this is percent positivity, just to show what's kind of happened in 2022. So as you can see, and Sarah Kidd showed this before, the enterorhinovirus activity really increased in August, September, October. This is being driven largely by EBD68 at our site, where we do surveillance for all our EVRV positivity. We got up to about 70% of our rhinoenteros were EBD68. So that was driving a lot of the respiratory disease during that period. And as you can see, as that went down, as Sarah mentioned, going into October, that's when RSV started to come about. And I'll show you the huge spikes of RSV we've been seeing. And then you can see here in the green that flu A is starting to peak its head. So you can see overall, the respiratory viral positivity is at a sky high rate. Lots of these viruses are coming back at once, and it's keeping a lot of children's hospitals very busy right now. So as I mentioned, the EBD68 outbreak that Sarah talked about has been closely followed by a very large spike in RSV. So this graph is kind of the yearly months of the year in RSV season, and the dark green line is this year. And as you can see on the right, it started to spike into September, but really is peaking in October into November. Look at that compared to previous years, and you can see just how early this RSV season is coming and how severe the rate of hospitalization is. So if you don't work in a children's hospital or don't see this side of things, many children's hospitals are trying to expand volumes. We have a tent outside our ER. We've shut down all of our ORs and are using our PACU beds as inpatient beds. It really is a kind of once in a career type surge of respiratory illness, and it came on the back of the EBD68 outbreak. The really concerning part is that it looks like influenza is starting to really come up as well. So this is the FluView data from CDC. And as you can see here in this top graph, this is percent positivity, and it's already getting up to 9–10%. This is mostly being driven by influenza A. Very little influenza B has been around since the pandemic. And we are starting to see flu hospitalizations increase. That's the red line here, at an earlier time than we would have expected from any of the previous influenza years marked here. As you can see, this is starting in the southern states as far as influenza-like illness activity, and is starting to move up to the Midwest and to the north. And unfortunately, we are already starting to see the first pediatric influenza-associated deaths being reported. The one silver lining of this is the majority of the influenza circulating is influenza A, H3N2, and it is a good match with the current influenza vaccine. So the message is to get your patients vaccinated as early as you can. It's circulating early, and we're hopeful that that will provide some protection, though influenza vaccination rates remain low. Lastly, COVID hasn't gone away. So these are COVID hospitalization rates in children. And as you can see here, it's starting to tick up. As far as what's circulating, this is still Omicron sublineages. BA.5 is kind of the darker teal here, which is still predominant, though it's decreasing as the Scrabble variants, BQ1 and BQ11, are starting to take over. So it's suspected that those likely will become dominant in the months coming into this winter. So we could be looking also at a COVID surge this winter. Fortunately, we now have bivalent vaccines, but as you can see here, the pediatric vaccine data is just abysmal. So I would say it's a pretty big success that we have 61% of our 12 to 17-year-olds through the primary series. That drops to about 30% in 5 to 11-year-olds and 4% of 2 to 4-year-olds. And then when you look at boosters, which have been available for some time now, about 2% of 12 to 17-year-olds and 0.5% of 5 to 11-year-olds. So a lot of work to be done there. So a lot of people are asking, as they're hearing the news reports in the media of just how busy and how at capacity all the children's hospitals are, what can we do to help? And really the message is to get vaccinated against what we can prevent. So that's the COVID-19 bivalent boosters, the seasonal influenza vaccine, and then the typical infection control recommendations: stay home from school and work when you're sick, wear a mask, wash your hands. And then I think especially with RSV, taking extra caution around young babies and infants and our elderly who are really susceptible. That's where the mortality comes from this disease. And then as far as AFM, we have a lot of patients on prolonged respiratory support, and it's really important that we take extra caution with these respiratory viruses that they're particularly susceptible to. On the public health side, the same ventilation airflow strategies for COVID work for many of these other respiratory viruses. So working on that in schools and public places. And then when we're at capacity, a lot of people are saying this is, the COVID surge for pediatrics in terms of respiratory volumes. You just want to make sure that we're keeping kids out of the hospital who don't need to be in the hospital for other reasons. So keep the cliff jumping for later in the year and try to keep your kids out of the hospital while they're at capacity. So I will leave it there. Happy to take any questions. This was a little bit of a side topic just to catch people up on what we're seeing on the PEDS ID side of things.
Kevin, this is great. Thank you very much for this good update. I am going to actually, why don't you keep the slides on the screen? I'm going to take advantage of being here in the microphone to ask a question about the slide number 4. In the slide number 4, if I understand correctly, you define this honeymoon period, and the prediction was in 2022 that there was going to be some increase in the number of cases. Did you take in account in that modeling the age of onset of AFM, for example, based on the previous outbreaks? And I think that that's an interesting point, because I believe that if there is going to be a honeymoon period, it's going to be a little bit more prolonged if we take in account the last outbreak was 2019, based on the age of onset in the outbreaks that happened in previous years.
So good question, Carlos. This is more modeling the circulation of EBD68 than AFM itself. So this was more respiratory virus circulation. It does say known periods of EBD68 and AFM outbreaks, but this was meant to be percent infection with EBD68. There was this lingering question, and we posed it in this paper of back in the old dusty polio papers that Amy will have shared with us. The older you were at primary infection, the more likely you were to go on to severity of illness and paralysis. And we do think likely we were going to see a higher age at initial exposure for those kids who hadn't seen EBD68 since 2018. And we didn't see the AFM surge that we were expecting. And I think that's the lingering question from this conference that a lot of us have been pondering. And I don't think anyone has a great answer yet. Obviously, we're all super grateful, and it's fortunate that we saw a lot of EBD68, but didn't see a lot of AFM and paralysis following. But I do think there's a lot of work to do to try to figure out why and what implications that has for the field moving forward.
Thank you, Kevin. Misha, you have a question.
Yes, great presentation, Kevin. Good summary. One question I have with the slide number 6. So this is all BioFire data, correct? Did you try to identify how many of these EVRV cases are EBD68? Do you have any estimation on that?
So it's a great question. Again, burden of disease is almost impossible to get at right now in the US with the way we do testing. So if you remember, we presented previously, there was a collaboration that we had with BioFire where they had this predictive modeling based on the melt curves for the EVRV primers. That went away when they upgraded from RP2 to RP2.1 to include the SARS-CoV-2 target. So they don't have the ability anymore to say within their rhino-entero target what percentage is EBD68. We really have to rely on our kind of sentinel site surveillance, which Sarah Kidd had mentioned the CDC is doing, and then a few of our sites, Iba Mostafa at Hopkins and our group, are doing testing behind the scenes for EBD68 amongst our EVRV positives. And like I said, we had about 8 weeks of sustained circulation. It maxed out at about 70% of our EVRVs being EBD68. But it's still really tough to get at overall burden of disease because none of the commercial platforms really test for EBD68 specifically. I do think we have the ear of some manufacturers. I think BioFire is actually very interested in the next iteration of RPP to have a EVRV detected EBD68 yes or no. So I think that would help enormously as far as the field in terms of understanding how widespread EBD68 circulation is, what the burden of disease is. And as we get to therapeutic trials for MATS monoclonals or vaccine candidates, that's really going to be a necessity to be able to know which of these EVRV kids is EV68.
Yeah, because like majority of these could be rhinovirus itself also, because rhinovirus numbers, if you showed in the other slide, the previous one, the big surge of rhinovirus itself. So it could be a mix of rhino or EVD68 and few of other enteroviruses probably.
Exactly. And that's a great point. And I think that gets forgotten a lot by clinicians, that every single fall we see a rhinovirus surge as school goes back and the allergy season comes in. There's an enormous influx of asthmatics that are rhino enteropositive that are typically rhinovirus every year. And then there's this, every other year pattern that on top of that, we have EV68. But it's really important, I think, to eventually be able to delineate what is EV68 versus rhinovirus.
And for with your slide number 4, was Carlos was asking, the honeymoon period is, and this is just thought I want to share. And we talked about it, Kevin, several times. If like, what if acute flaccid myelitis is a very small number of cases? It could be antibody dependent enhancement by combination of different enteroviruses. Is it possible these kids during the time they have not seen any other enterovirus infection also? So when they get EVD68 infection, they do not have that kind of antibody enhancement or that kind of inflammation, or they do not lead to that kind of conditions like acute flaccid myelitis. So they do not have yet that second infection that need to trigger that condition yet.
I think that's a really interesting hypothesis. Like I said, I don't think any of us know for sure what happened this year or why, but things like that I think could be tested further. I know Matt Vogt has thought a lot about antibody dependent enhancement and hasn't seen kind of evidence of it in his humoral immunity studies. I don't know if Matt wants to speak up, but I mean, I think we pursue all possible explanations and try to figure out why this was. My hope is at least we got a lot of immunity this year because a lot of kids saw EV68. So hopefully we'll push off the next outbreak for a while. But I think it'd be really great to be able to answer this question of why didn't we see a lot of AFM this year? And I don't know.
Yeah, I agree. Anything's possible, but I mean, neurons don't have FC receptors, so I don't understand how that would work. That's the main thing that I think of. But again, who knows? Why I have zero clue why we didn't see AFM this year.
All right, thanks.
Hey, Kevin, may I ask you a question? So if you look at the epidemiological data for the prevalence of D68, you can see it's peak in the fall and decrease in winter and early spring. My question is about the superinfection between D68 and other respiratory viruses. If you look at the COVID data for patients with COVID, co-infection with COVID and other respiratory virus, the occurrence is not high. How about for D68 patients? Does it mean if the patient in the season with other respiratory virus, the co-infection with the D68 will be decreased?
That's a good question. I don't know the answer for respiratory disease cases of EV68 and co-infection. I know the CDC will have pretty good data from the AFM cases because those are pretty well characterized. From our own data, I haven't seen a lot of co-infection. I know we in particular had one EV68, EV71 co-infected AFM case from 2018, but I don't remember a ton of co-infections. I don't know if Sarah or Janelle want to speak from the CDC's data set there. And as far as seasonality, I think the late summer to early fall circulation is thought to be driven by dew point temperature. So a combination of humidity in temperature in temporal areas, but that goes away as you get down into the southern states. So it tends to stay away from most other respiratory viruses besides rhinoviruses because RSV is typically not here and flu's typically not here by then. But Sarah and Janelle, do you have any other co-infection data from the CDC data set?
Sarah, go ahead. Yep.
So CDC lab would normally just test for rhinoviruses, enteroviruses. We do have a couple of instances of co-infection with a rhinovirus and another enterovirus or two rhinoviruses. It's not common. I'm just trying to think if we have any data on other respiratory infections like RSV or COVID, I think we do get that with chart abstractions, and I don't know if we've ever quantified it, but it's not common. Probably because of the seasonality that you were referring to, Kevin.
And Ben, I do think also it's not because we're missing things because we're not testing, because we did do some next gen sequencing of the nasopharyngeal samples back in 2014. And with that question, is it like something else that we're not testing for that's also there? We weren't finding anything consistently along with the D68 that was found in those cases. But it's a good question. And if anyone else has hypotheses or questions that we could look into the data from this year, definitely let us know. In addition to the natural history study that was going on this year, we had a great shedding study that was really successful. We enrolled around 50 families that were EV or RV positive and swabbed them for 21 days, got blood and saliva before and after. So I'm hopeful that we can dig into some of the humoral mucosal immunity questions and the shedding curve and things like that, so we can get better data out of this out.
Thank you.
Kevin, and not only for Kevin, but for the virology group, do you think that the circulating virus that is present at this moment in the United States, as well as the B3 that is circulating in Europe, is the difference for the lack of cases? I mean, from genomic analysis of those viruses, do you think that there is any clue we are now experiencing AFM cases?
No. So my first publication demonstrated that neurovirulence is not an evolving trait, that if you ask the virus to be able to infect cells of the central nervous system, they will all be able to infect cells of the central nervous system. My work was then recapitulated by Ken Tyler's group. It has to do with the host.
The host, yeah. Great, thank you.
So Amy, what do
other...
you think changed with the hosts this year?
What?
What is your hypothesis of what happened with the host this year? If it's not virus, we saw a lot of virus going around. What was different about the kids? Do you think it was, different immunity background?
It could be different immunity backgrounds. It could be different environments. Yeah, it could be a ton of different things. They could be, while historically for polio, when we moved into good sanitation, we pushed polio into older adults, into older children, and they had more ability to develop paralysis. We know that the penetrance of polio is 100,000 times higher than it is for EVD68. So like approximately 1 in every 100,000 children who've infected with EVD68 do we see one with paralysis. Whereas for polio, depending on the type, you're talking between 100 and 1 in 100 and 1 in 2,000. So like for type 2, it's 1 in 2,000. For types 1 and 3, it's like 1 to 100 and 1 to 500, somewhere in that range. So just the penetrance of the virus is completely different. So yeah, it's really not the virus. It has to do with the host completely. And it's not clear exactly what it is. As I said, it's going to be complicated, but you can just see that the penetrance of these diseases is very different.
So Kevin and Matt, do you think that the immunological profiling of these samples that you obtained this year are going to be helpful in many ways to characterize some different specificity of the antibody response versus the historical AFM or potential future AFM?
Certainly. I mean, we only started collecting this year, so the future is where our power lies. I mean, yeah, we could dig up old samples out of freezers and compare, I suppose. But yeah, I think given the absurd amount of EVD68 infections in children, we have enough people we've enrolled that we will have kids who were naive, or even if not naive, it had been a long time, when we got sample 1, and then they will have been, boosted or infected or whatever you want to call it during the year. Question for Sarah and Janelle, I guess, does the NVSN is their workflow where they do one of these multiplex panels first and then they type for D68? Do you know? And the reason I ask is going back to the questions about co-infections, but maybe to flip it on its head, are people who are not getting AFM, which would be literally every D68 detection from the NVSN, like, do they have a high incidence of co-infections? Because that data may be available to you.
Yes, that would be, yeah, thanks for filling
Okay.
in, addressing
Okay.
that question. Yeah, the NVSN, we would be able to get data on co-infections, because
Yeah.
they monitor multiple respiratory viruses.
And just anecdotally, I'm sure Kevin will back this up because he sees a lot more patients than I do too. You know, there's so many co-infections in these kids coming that are jamming the hospitals right now.
I was trying to prompt Janelle, had a story of a friend's child who just tested positive for
Oh,
5.
That's
5
very
on the
impressive.
respiratory. Yeah.
That's very impressive.
yeah, it was crazy. Yeah.
Bernardo, you have a very important clue for us.
No, I don't have an important clue. I just want to be careful to follow up from Amy's comments that we say that the virus has nothing to do with it. I will say I disagree with it. I mean, I think the jury's still out. I think we want to be careful and be open to different options. I think, Emma Hotcroft's paper showed there's some really interesting differences with the A2 clade, where they saw some different levels of in the older populations, and you didn't really see in the other clades. So I think I think we all know, right? I mean, what we know is that we don't know. And I think we don't want to close doors because of an animal model. I think that there's a lot that needs to be teased out. And I just don't think we have answers. And D68 is not polio, and polio is not D68. I just want to be, in my opinion at least, and Amy knows NROs way better than me, but I think that the jury's still out. And I just don't want to say it's not the virus, it's not the host. I think it's probably a little bit of everything, and we just don't know. And once we know, we're going to be like, how did we miss that? Right? Once you see it. So.
Thank you very much. Well, thank you everybody for being present for the meeting. This was fantastic. Thank you to the CDC group, Sarah, Janelle, Adriana, Kevin, and Jelty for